So first of all, I would like to thank the organizers and the chairman for inviting me to share our data with you. And I will present in the first part my field because we are going to move on to hematological diseases. So it's quite different from the different presentations you had already this morning. I'm based in Marseille, the University Hospital. And for the research unit is about neurology, and I'm head of the Laboratory of Biochemistry and Molecular Biology. So it will be molecular data, and I will introduce you the primary myelo-proliferative syndromes. Maybe you have heard of them. About the MPN negative, which are negative for the Philadelphia chromosome, which at the molecular level they are negative for BCR-ABL, and we know that we have a personal drug which called anti-tyrosine kinase inhibitors, and so if they are negative for BCR-ABL, we have three different diseases here. Polycythemia vera, it's an increase in red cells, and in this disease, we found a mutation for JAK2 tyrosine kinase in 95% of the patients. So I will remember that 20 years ago, when I started, it was exactly the same for BCR-ABL. Now we speak about chronic myeloid leukemias only for patients bearing the BCR-ABL. So it will maybe be the same thing in a few years for this disease. The next one is essential thrombocythemia. It's an increase in platelets. And half of these patients are mutated for JAK2 gene. The third disease is primary myelofibrosis. And again, half of them bears the JAK2 mutation. So we can ask if there are other genes involved, and the answer is yes, of course. Very recently, it's 2 years ago now, almost, even less, there is a mutation found in the CALR gene that I'm going to talk about. And we knew already there is the JAK2, but also PML, gene alpha, which was mutated in 7% of the cases, which is quite rare. So in less than 10 years, 3 exclusive mutations have been found for these patients, explaining 90% of the patients. So let me introduce you the CALR gene. So, and the protein, here you have the protein. With the 3 different domains. The mutations are all located in the C domains for the, which is involved in the transport of the calcium, but we don't know yet. We don't have enough research about the consequence and why there is a link between these mutations and the hematological diseases I introduced to you. In any case, patients are treated either with classic drugs or with targeted therapy against JAK2 inhibitors. And we see that JAK2, you can see, is below action and actually also acting through patients, the mutation of in the exon 9 of this cartoon of these mutations. So you have 2 members representing or an addition of 5 nucleotide rare. Is there any involvement in the prognostic of the disease for the patients bearing these mutations? The answer is yes, because you can see what happens to the patients. This is a survival curve when we didn't know the mutations. Then you can see that it's worse if there are mutation JAK2, the first mutation none, compared to non-JAK2. And for the calreticulin gene, the prognosis is better than JAK2. And then the patients who are negative without mutation for the 3 genes, so neither JAK2, calreticulin, or MPL. And you can see that the prognosis is worse for these patients. So you have the number here on this slide. Well, then, if we look inside the calreticulin gene, I told you that the 2 main mutations, and we can see that for the survival, the 2 main mutations do not have the same prognosis, because this is the survival curve for type 1, and the survival curve for type 2 is the same as JAK2 mutation patients. So clearly, these diseases, essential thrombocythemia or primitive myelo-proliferative fibrosis, can present 2 phases we didn't know 10 years ago. Because if they are mutated with JAK2, they have an increased risk of thrombolysis. If they have a mutation of calreticulin, they have an increase in platelets. So now move on to our work. We are part of the West PACA region, the south of France, for genetic abnormalities in cancers. There are 28 platforms in my country. So running something like 60,000 tests per year for all cancer patients, I would say, not only the one I presented here. And so in my lab, what we do perform is a real-time PCR detection for the JAK2 mutation, which is only located on 1 position. You change a valine for an histidine. And for the calreticulin gene, it's a different technology. We screen, the first screen is high-resolution melting curve. And then, if positive, we can sequence by Sanger, classical sequencing, if positive. Here are data. So out of 200 patients, we had the same proportion of patients, of positive patients, 68 for JAK2, 32 for calreticulin. And what was our surprise is that we looked at the age and we realized that the age association with the mutations is related with sex without explanation so far. Because as you see, we know that, calreticulin patients are younger than JAK2 patients, JAK2 mutated patients. Okay, but actually what you can see on this slide is that this is true only for male, the green bars. And we just don't know why. But that's surprising. So our conclusion is that the WHO guidelines were written in 2008, and they should be revised because we discovered these mutations now. And diagnostical tests just moved before as only an exclusion diagnosis, because we didn't know the mutations. Now we can speak about personal medicines, and the physician can apply a better treatment for each patient. So we find also similar mutation and implication around the world. That meaning that the studies from China or Asia are comparable from the one from Africa or Europe or the States, exactly the same. So I spoke today about 3 mutations, but there are much more, and apparently they can have a prognostical interest. We'll see. And also a very active research area is a treatment about targeted therapy. Okay, but there are a lot of questions. What is the physiological role of this calreticulin gene and protein? We don't know. Why all the mutations are located only on exon 9? We just don't know, which correspond, I told you, to the C domain bearing the calcium. Why is the difference of age? I told you about it. And we would like to build a network, a Euro-Mediterranean network, involving this country, so Spain, and I will meet people from Valencia about that. France, Italy, but also Italy, and Lebanon, and Tunisia will join us on these diseases. And now I would like to thank people from my lab. So Caroline Bonfoy is a young physician. She is doing a thesis. Nathalie Beaufils is my engineer. And 2 colleagues, Dr. Amin Ben Laswed and Dr. Fabricius Siglio, but also the National Cancer Institute Agency in INCA and the PACA region. Thank you very much for your attention, and I will take questions.
Questions?
Thank you. It's quite nice talk. This mutation in CALR gene in these patients, are they unique ones or are accumulated with another genes? I mean, in other words, has to have another mutation in other genes, or this would be enough to provoke the disease?
Just to be short, we don't know, but actually it's enough to make the disease. But apparently other mutations are important for the prognosis, okay? Because actually so far we knew about 1 disease, and knowing about these mutations, what it appears is that you have 1 disease, it's the same phenotype when you look at your microscope, but actually, depending on which gene is mutated, the prognosis is different. But to answer your question, yes, if you have only calreticulin gene mutated, that should be enough. I didn't see so far any publication. We know that for BCR-ABL, but it's known already. We know that only this BCR-ABL in mice, for example, it's enough.